midnight, one that's due on Wednesday at midnight. One of them's a case study on the immune system. The other one is a journey through the lymphatic and immune system. So you're going to encounter all those things that we're learning about and that kind of thing. Okay? So, Jamie's going to let me know if it doesn't pop up for the case study for some of y'all. So, with this initiating the immune response, we've got all the players in place now. We know that we are looking for major histocompatibility proteins or complexes that are found on the surface of our cells. Right? We're looking for self proteins is what I like to think. And when we find something that's non-self, what we do? Kill it and eat it. Okay? And that's what the macrophage does. Okay? So the macrophage, and Alex had a lymph node club going in the lab today, and all these players that were involved in this. And we said that this macrophage usually hangs out at the doorway to the club, checking people getting in, right? Stamping wrists, checking things.
Sure.
Okay? So that works pretty good. Okay? Now there's an issue. T cells, okay, can only deal with something that is presented to them from the cell. Okay? We talked about an antigen presenting cell before, right?
Yes.
Okay? And the macrophage can do that. It can be part of that. We can identify what it is from that antigen presenting cell. The T cell can do that. Okay? So it will bind to it. Now, there's two different kinds of binding. One binding is to activate. The other is to destroy. Okay, so this is the activating side of binding to an antigen presenting cell. Okay? And in order to do that, we said that foreign particles were taken in, partially digested, and then placed on the outside of the surface of the plasma membrane. Okay? 2 times this happens. One, if you're infected, you become an antigen-presenting cell. If you're infected, hey, they got me, this is what's got me. Okay? And the second one is if you're this macrophage presenting it. Okay? So,
Can you explain the second one?
what?
The second one.
If you are the macrophage and you're trying to activate the immune system, you want to show them what you have found. Okay? So that's this antigen presenting cell. Okay? 2 different kinds. One, a macrophage trying to alert the immune system as to what's going on. Another cell doing basically the same thing, except also saying they got me, I need to be killed. But these are inside me. We're still presenting it to the immune system. Okay? So, macrophage that encounter major histocompatibility complex proteins that aren't right, which we would then really be an antigen. Okay? Any antigen, then it is going to eat it. Okay? It says, or a virus that's infected with that cell will stick it out on the surface, right? Okay? So both of those systems work. And they release interleukin 1. Okay? Interleukin 1 is the call to the immune system cells. Okay? Now, the cell that is most often involved in this is the helper T cell because it's able to initiate the immune response. That's its function. I'm able to initiate the immune response. So we need to activate the helper T cell. And basically by activating it, we are showing this improper protein that is in our body to it. Okay? This is foreign. It needs to die. Okay? So at this point, the macrophage may have already killed it, right? And stuck it on the outside and said, here it is. Or the infected cell, whatever it is, may have done the same thing. They're making viral particles inside of me. We need to kill me. Okay? So, interleukin 1 is the call to the helper T cell. we talked about positive chemotaxis before. A little bit of blood in the water brings the sharks, right? Okay, so the same thing here. A little bit of interleukin-1 brings the helper T cells over. Okay? And what they're gonna do is we're gonna activate the helper T cell. And now that it's activated, the job of the helper T cell is to talk to both sides of this immune system. A T cell side, which we call the cell-mediated side. The humoral side, which is the B cell side, right? Okay, so B cells and T cells are going to be involved in this, and the helper T cell can talk to both. That gives us the cellular response of T cells, the humoral response of B cells. Okay, so helper T cells are activated when they come in contact with this new shape. Okay, and now their job is, okay, I gotta find it. I'm activated, let me go find. And if all of y'all are the T cells that we've got on board in our immune system, and I'm the helper T cell and I've been activated, here's the shape I'm looking for. Any of y'all match that shape. So I'm gonna go around and try to find the cytotoxic T cells that we've already got out there that know this shape. Okay? I'm also talking to B cells at the same time, trying to figure out if they've got the shape. Do you have an antibody that matches this shape? Okay? I may go through the whole group of them, but what's an easier way than me traveling around to all of the different T cells? Let's have y'all come to me. Right? So let me release some interleukin-2 and bring all of y'all, and y'all just line up and check and see if y'all are going to work. And I'm sitting here going, I'm activated. Do you have the shape? Get out of here. Do you have the shape? Get out of here, you know, back until we find one that works. Now here's the deal. It may just work a little bit, but that's a little bit of help, right, at this point. We can make another antibody later on that's better, but if we've got one that works 25%, that might be worth investing a little bit of time in, right? So we make a few of those. If we don't have one at all, or we have one that doesn't work very well, we gotta make it. Okay, we gotta make it. So, helper T cells secrete the interleukin-2. This is going to stimulate the production of cytotoxic T cells on the T cell side. Meaning, if I find one, okay, maybe Halima is the one that matches. Now we need a bunch of Halimas, right? Because she knows she's got the right one, and there's going to be cells that we need to knock out. She's a cytotoxic T cell. She's going to go out and start destroying anybody that's doing this. Okay? So we make a bunch of Halimas. Okay? And we turn her loose, going out looking for cells so that we can destroy the factories. So, anybody that has it sticking off is going to die. So this is what this looks like. Here's our macrophage gobbling up the viruses that it found at the doorway to club lymph node.
Oh, lymph node, that's right.
We found somebody trying to sneak in that was underage. Okay? We ate them. Brought them in, presented their ID. Okay? This date is wrong. Anybody that has this date or below it needs to die. Weird club. Okay? Let's show this to the helper T cell. How do I show it to the helper T cell? I release interleukin-1 and the helper T cell comes to me. It goes, gotcha, big guy. Good job. Okay? Let me go see if we got it. So now, release interleukin-2, and that is looking for one of these. A cytotoxic T cell that has the ability to match to that. Okay? Now, the same thing can happen if we find the antigen-presenting cell out in the body, right? If it's out in the body, it's not being presented by the macrophage, it's being presented by a cell that's making more viral particles now that cytotoxic T cell goes, oh, they're back. Okay, they're back, and I'm going to do proliferation. I'm gonna make more of me, because if they're back, they need to die. We're gonna kill this one, we're gonna make a bunch of this. Now we've got a bunch of cytotoxic T cells, a bunch of Halemas out there that anybody underage that's being shown is gonna die. Okay, we're gonna kill that cell that is being exposed. That takes care of the factories. Okay, that takes care of the factories. Now, we haven't done anything about the individual viruses running around in our body. All we've done is knock out the factories. And that's what the whole T cell side does. Okay, it knocks out the factory. So now we've got to work on.
May I ask a question? If I go back to the step, last slide, sorry. I want to get two things clear. I will talk about the antigen-presenting cells. And you said that's the one that the essentially says they've got in right, and shows and it says that they've got in at the helper T cells. Interleukin 1 is presented. In the upper case where we have the microphage, at the point where the T cell receptor, or at the point where the underage guy that was killed, is that when it was being presented outside, you say that antigen, is it also an antigen presenting cell at that point? So whether it is the microphage or the
Infected cell.
infected cell, it's the same antigen presenting cell.
Absolutely.
So when it's there, interleukin 1 is released. Is it you get the helper cell before interleukin 1 and interleukin 1 before the helper cell?
We've got helper cells running in our body right now.
So when you have the helper cell, is that when they call on the interleukin 1 or before?
When interleukin 1 is released, the helper T cell goes over and identifies what the shape is.
Oh, okay, interleukin 1. When the macrophage is released outside as an antigen presenting cell,
Okay?
so what happens there?
Then it releases interleukin 1.
The macrophage will release interleukin 1.
Yes. Because it's got something to show the helper T cell.
Okay, so the helper T cell will come. When you identify the now goes releases interleukin 6. So is the helper T cell that releases interleukin 2, right?
Yes.
Okay.
Okay.
Then it now proliferates its own kind to be able to fight
To be able to knock out that cell, that's all the antigen presenting cells that are shown.
Oh, okay.
Okay. So now we've got the B cells. Okay? And B cells, the cool thing is they're not doing, the B cell itself doesn't have to do any recognition or anything like that. What it does is make an antibody that it's going to release that'll go out and mark it for destruction. Okay? So the B cell doesn't have to do anything with checking IDs or anything like that. It just makes the antibody. Okay, and that's what it says right here. Doesn't have an ID system of its own. Its job is to make the antibody. Okay? And the good thing about the B cells is they can knock out the individual viral particles in the body. Okay? And the way we think about it is a lot of these viruses that we get, we never get rid of all of them. You still have the virus. Okay? So, if that's the case, oftentimes we continue killing this virus throughout our life, right? Because we have the machinery to do it. Okay? So, a good example of that is chickenpox. Okay? What happens when you get older and you get stressed is the chickenpox virus hides in our nervous system, and we don't have good immune system in our nerves. Okay? They're not supposed to get that far. But this one hides out in our nervous system, and when it is able to proliferate due to there's some kind of stress in the body, the chickenpox virus will go crazy, get in the nerve, and it'll replicate in the nerve cells, and we end up having
Shingles.
Shingles. And shingles we think about as usually an older, like y'all shouldn't, but sometimes it happens. Right?
What do they look
Sometimes.
like?
What?
What do they look like?
Little chicken pox.
Little dots?
Little dots on your
Very
face.
painful.
Okay?
So, and they're painful, yes. If they're on your torso and your shirt rubs against it, it is very, my dad had to just go around without a shirt for several days because it hurt so bad. My wife got it because she went to the dentist, and my wife, there's her mandibular joint back here doesn't allow her to freely open her mouth really, really wide. You know? So they have to put those blocks in there to keep her jaws open while she's having dental surgery and that kind of stuff. When that happened, that was enough stress, mechanical stress in this case, to have the chickenpox virus come out in her, y'all remember the trigeminal nerve? So it came out right here. She had pox that went down here. She had it that went right to the edge of her nose. And she had it that went above her eye. And they had to get a eye ophthalmologist involved because if it got in her eye, that could be really bad. So she had a team of doctors working on this chickenpox virus that's running loose in her body. Okay? And they gave her antivirals and that kind of stuff and knocked it out pretty quick. Okay?
Is shingles contagious like chickenpox?
Yes, it is the chickenpox virus. Okay? So individuals that have not had chickenpox could get it. Okay? Now, I've had chickenpox, so it was not an issue. I just make fun of her. Right? Because she had this thing growing on her face like this. Right? So every time she would come in the door, oh, okay, sorry. She didn't like that at all, y'all. I pretty much just stay in trouble. Okay? So, same thing with me. I told y'all I had hepatitis. Could I still have the virus in my body? Sure. Right? Sure. And that's why they don't want me to give blood. Because is there a chance? Yeah, there's a chance. Okay? So, lots of these things. And that's the same reason when they do the Valtrex commercials. What's Valtrex for?
Herpes?
Herpes virus, right? And they say, they're real happy about it too. Valtrex controls my outbreaks, right? Because the virus doesn't go away. Okay? So, lots of those. It's the gift that keeps on giving, they say. Okay?
Herpes is like luggage. You never get rid of it.
That's right. And somebody else can get your luggage. Okay. So, antigens, okay, they're going to enter the B cell by endocytosis. They're going to get processed, and we're going to build something that will fit that marker. We're going to build something to match that marker in the B cell. So here's the deal, and we learned this. The RH mama, RH negative mom, had to see the RH positive blood before she would make antibodies to the RH positive blood, right? So the first baby was fine, second baby wasn't. Okay? So once that is seen, once that antigen is presented, right, it gets taken in by the B cell. The B cell goes, okay, I'll make antibodies to this. Okay? And now we're ready for the next baby. Okay? Or any other RH positive blood. Okay? So, helper T cell is going to do that. It's going to help recognize the specific antigen. So we had to go through this whole process of building this cell. Okay? So, antibodies are just proteins, y'all. And that's cool because we know how proteins are built, right? We know that there's some DNA that gets converted into messenger RNA that gets sent out to the nucleus, out to the ribosome. A gene is involved in the coding of this protein. That messenger RNA goes out, and we just simply put amino acids in the correct order and peptide bond them together, right? Easy peasy, lemon squeezy, right? So, these antibodies come in different classes, though. They've got different functions. They're called immunoglobulins because they are part of that globulin fraction that's floating around in our blood that we learned about, those plasma proteins. Now, these are small enough that they can go across the capillary bed. Remember how those antibodies affected the baby's blood because there actually was mixing here. The blood doesn't mix, but these small proteins, these little Y-shaped antibodies can go across. Okay? So we've got these immunoglobulins. They call them IG and then a letter. IG whatever, okay? So the IgM is the ones that are the first to be released. The first time, and we can do this, we can say to somebody, you now have COVID. You are expressing IgM antibodies, okay? During a secondary response to this, you do a little more sleek antibody. It's kind of easier for us to build and send out a bunch of. It's an IgG. And that's, I was hunting with my buddy 2 weekends ago. And that's what he does. He samples the blood, all the blood that comes in from the United States that, not all of it, but a lot of it that comes into the United States that is being donated. They take a small sample of that and he runs markers to identify what that blood possesses antibodies to. Okay? Or what that person had, because there's a chance that could be bad blood, right? There could be a virus, there could be a blood virus that the person doesn't even know about that's in it. So his job is to screen this, and in doing so, he can look for all kinds of stuff. So one of the things he's doing right now is the COVID stuff. Does it have COVID antibodies, and is it IgM only? What would that mean?
It's infected, COVID infected.
Or that person was vaccinated against COVID, and that's it, right? Or IgG, what would that mean?
He wasn't vaccinated.
And
Got it.
has been exposed to COVID. And now we're fighting. So that's how cool that is, right? It's pretty cool that we can do that. And I said, so if I gave you my blood, because I've been vaccinated and I've been sick a couple of times, but nothing like I thought I had COVID. He said, oh yeah, we can do that. I was like, okay. So at some point I'm gonna have to send Phil, Dr. Phil, okay? And when you hear me talk about my buddy that's got a big head, it's the same guy, okay?
What's the difference between primary and secondary response?
First time you get it versus subsequent infections. Okay, IgD, and I'll show you this, it is the ones that we find on the B cell. It's the receptor for antigens on the B cell. And what that means is once we make these B cells, we're gonna go ahead and have these antibodies sticking off, because if they run into something and bind to it, we know they're back, we know this one works, I know I better make a bunch of meat right now. Let's start this process, okay? So it's an IgD antibody. There's an IgA that is found more in external secretions. So we have antibodies in our saliva. We have antibodies in the fluid in our eye. Mama makes breast milk to give it to the baby. These are all external secretions, okay? And these are ones that we can send across and we can give to other individuals through that form. And then we have IgE. And these are the ones that work along with a mast cell in our connective tissue, typically. A lot of times mucous membranes. And it's what's going to facilitate that allergic reaction that we get to this, okay? It's gonna facilitate histamine release, and histamines cause vasodilation, which is gonna cause more blood flow into the area, which means it's gonna turn red, and it's gonna leak out more fluid, so we're gonna get some swelling. Our eyes are gonna water if that's the case. Our throat's gonna get more. So there's people that can walk into somebody's apartment and go, you've got a cat. Like they just set foot, right? And it's immediate, you feel it and you know what it feels like. Your eyes start watering, right? Your nose gets itchy and then it starts producing more fluid and that kind of stuff. Okay, so that's where these are gonna come in. So 5 different classes of these. For us, I'll be honest, these 2 are the big ones, okay? These 2 are the big ones. This one's important because of the allergic reactions that we can have here. These other 2, IgA is cool because it's in the external secretions, but Okay, so here's the way the bees are gonna do it. The B cells are gonna do it. Same system, got a macrophage that encounters something foreign, it eats it. Processes it, puts it on the outside of its cell, being that antigen presenting cell, and it releases interleukin 1. Interleukin 1 is the call to the helper T cell. Hey, come see what I got. Helper T cell comes over. I got you. This is what's in our body. Let me see if we got somebody out there that knows how to kill this first, right? If we go through, it's going to release interleukin 2 to call. Did I already release interleukin 1?
Yeah.
Yep.
Okay, I already released interleukin. So the helper T cell now is releasing interleukin 2. Y'all come check. Let's see if we got somebody out there that knows this shape. If not, we're going to have to build it. How long is that going to take? 4 or 5 days maybe to build this and get it up and running and get enough of them out there so we're fighting the fight. Okay. If we've got it, we identify it. I've activated it. I bring a B cell over and go, hey, you're the one. You make these. You make these antibodies to this. You're my guy. And he goes, I know. I've actually encountered them already myself. Right? I got some already bound to it. Okay? Now the B cell can do the same thing. It can bring it in, process it, make the antibody, have this because it's been exposed to it. Okay? But the helper T cell says, dude, turn you into a plasma cell. Now, a B cell and a plasma cell are similar, but not the same thing. A B cell turns into a plasma cell when its job becomes to make and release copious amounts of antibodies. Okay?
Say that one more time.
Its job, a B cell gets activated to become a plasma cell. And look at what we've done. Went from a B cell, they're not showing us the rough endoplasmic reticulum, right? To, hey, we got this rough endoplasmic reticulum. What's the rough part of the endoplasmic reticulum? Ribosomes. Okay? And if it goes to the fixed ribosome, we know it's for external release from the body, right? It's not a free, it's inside. It's not going to be used inside the body, it's going to be used outside the body. So we make tons of them, and the Golgi body wraps it up for us in these nice big transport vesicles. His job is to go over here and dump them as we're making it.
Excellent.
Dump as we make it. Dump copious amounts of these antibodies. Okay? Hundreds of antibodies from one cell that hit the fluids, right? And remember, this B cell more than likely is in a lymph node, right? In that medullary region of the lymph node. So as we're making a bunch of these B cells, that lymph node gets bigger and bigger and bigger, swells up. Okay? And with this swelling, we feel that, but we're also releasing it very close by where most of the infected cells are, right? Which would make sense. So, when it is a plasma cell, its job is to make and release. That's different from the B cell's job of just hanging out, waiting. Okay? What the B cell then, as we do this differentiation, we also make memory cells. And what's on the outside of memory cells? The antibody. So we know what your shape is for. Okay? We know what your shape is for. You're designed to make this and only this protein that's your job. So it sits there waiting and waiting and waiting until, bam, they're back. Convert from a memory cell to a plasma cell, release a bunch more antibodies. Okay? That time frame is so short, we rarely know that we were sick. We rarely know that something was wrong. We may feel just a little rough that one day. Okay? But then we wake up the next day and everything's fine.
So like my girlfriend the other day, she had a swollen lymph node on the back of her back, but she never had a back.
Right. Absolutely. Absolutely. And if it's the one back here, kind of where the occiput is, that's the one I always said, that's the one to get my wife when she has mono.
So be careful.
On the back of her neck. On the back of her neck.
Yeah, there's several.
And
So-
mine could affect the spleen, right?
Can affect your spleen, yeah. Okay, so this side, what we're doing is we're chasing down the little particles, right? Those that can go out and infect other things, affect other cells in our body, or, you know, there's that time in the sickness where the virus is winning so much that we have so many of these viral particles inside of our body, we're contagious now, right? Okay? And they say this with COVID. There's at a certain point, you're no longer contagious because we're winning the fight now. Okay? We've knocked down the viral load so much in our body, it's harder for us to transmit it to somebody else. Okay? So, when they're winning, we don't have our stuff together. We'll set this word right there. Okay? So these antibodies remind me of hemoglobin a little bit. 2 polypeptide chains. Okay? There's alpha and a beta. Same thing we've got in the 4 polypeptide chains that we've got in the hemoglobin. 2 alpha, 2 betas. Okay? So both of them have 4. 2 light chains, 2 heavy chains. The main part of it is pretty straightforward. I'll come back to that in a minute. So it looks like this. And notice that the blue part, the heavy chain, is pretty consistent. And the red part is a lot of what we can change. The light chains are what we can change. Got them. And y'all can see that that kind of shaped like a Y, right? So they did a pretty good job of showing us this Y. And both ends of the Y have exactly the same shape to it. So either side of this antibody can bind to it, where it says antigen binding site to it. Any side of it can bind to that. What are each one of those little bitty blue circles and red circles?
Amino acids.
Heavy chain and light chain?
What are the individual little blue and red circles that make up the heavy chains and the light chains?
Globulin proteins?
Now, this whole thing is a protein,
Amino acids.
so they're all amino acids,
Amino
right?
acids.
And that all a protein is a bunch of amino acids strung together. It's got a little carbohydrate in the middle. Okay? So
you said the blue is what amino acid?
each blue circle and each red circle is an amino acid. Okay? All right. So here's the deal. It says it's estimated that each T cell can make between 10 to the 6 and 10 to the 9 different antibody molecules. It means we make that many proteins, right? Then why don't we do that? Why don't we make every freaking one and have them ready to go in case we get anything? Well, genetically, we don't have that many genes, y'all. We don't have enough genes to do all that. So, we came up with a system where there is a suite of genes. So we've got several genes that code for shapes. Okay? Several genes that code for shapes. And when we code for the shape, we can put these in different orders and make different shapes based on the order that the gene is put in when we're making the amino acid, the antibody. So let's say one is this and the other is this shape. So I can have this going on, right? Or I can have this going on, let's say. Okay? So what they do is they have these different genes, suite of genes, and we just put them in different orders to come up with a different shape. And when we do that, we're able, they call this somatic rearrangement. Okay? We're rearranging the genes into a proper shape that matches. Okay? Into a proper shape that matches. When we do this, we make about 19 million different possible antibodies. Okay?
Cool.
And remember,
I may not make exactly the same antibody that you make, right? Because if that antigen is big, I may pick something that you didn't pick. And what if I got a different version of the virus than you did? We got all these different COVID strains now, right, that are a little bit different. What if I make mine a little bit different? Might mine work better with something later and yours doesn't work at all? So everybody's immune system in here is unique when we think about it. We've all had different things. We've all had to take the task of doing it ourselves. Okay? And sometimes it works really well. Other times it doesn't work as well. Okay?
So we all make these like composite shapes just all the time for no reason.
No. Only if we see it. And that's the
Oh,
deal.
I was gonna say, so only if we see it, we arrange it to,
Right.
but, so we have just the ability to make these different shapes.
19 million different shapes.
Okay.
But yeah, and that, the question I asked earlier, why don't we just make all that? We wouldn't have enough room in the body to put all these cells too, that are just hanging out waiting to happen. You know, when you think about it, how many different times have you really been sick in your life? Maybe 100, let's say. Well, that's, you know, but then there's a few times where it became very important for you to make antibodies to things. Okay? Like lethal, if not. Right? So, you know, if somebody that messes with rattlesnakes, good example of it, something messes with rattlesnakes. Okay? And if I was going to be a preacher and I was going to be one of them snake handling preachers, right? I would give myself every day a little bit of snake venom. Just a tiny bit that wouldn't kill me. Just, right? And my body would make antibodies to it, right? Antibodies to it. And as I gradually increased that dosage, if by chance I got unlucky and got bit like that one dude who got just bit right here in the face, I saw that on, freaked me out. He did die too, okay? You've got a shot. You've got a shot at it this way. So there's lots of people that do this kind of thing that are in specific trades where this could be the deal. But knowing me, I'd get bit by a completely different snake, right? Different snake species, and it'd just kill me dead, right?
But isn't that what they do?
I'd be messing with rattlesnakes and cobra biting me on the rear end, right?
Isn't that what they do with like horse's blood? They do antibodies that way.
That's how we get the snake venom.
Yeah.
So when we give anti-venom to somebody, we're giving horses antibodies to the person, so it'll latch onto it and destroy the protein in the venom, denature it. All right, our bodies will take care of it. And they used to say you could only get the anti-venom one time because it's a horse's protein that you've put in us now, and we're going to make now antibodies to the antibodies. But that hadn't shown to be proven as much lately, where people have had to get it more than once and it's worked. So I don't know what's going on there.
Now, is this to be true with, or is it different with identical twins?
What would you mean?
Well,
Would they have the same immune system?
right. I would say no,
if
right?
they caught everything the same, and that's probably not very likely. You know, one may go to the bathroom one day, and the other one didn't go in that bathroom, and there was something in the bathroom. I don't like that. It's spooky. Okay, so this is the making of the antibodies. Okay. All right. Now, it says there is tolerance here. The immune response does not respond to the animal's own tissue. Now, when we start out as an embryo, we are learning what our own tissues look like. And remember, every one of these tissues in our body have different protein markers on them, right? So as we go through embryogenesis, the making of all these different tissue types in our bodies, there's going to be new ones that come online, and new proteins are going to be utilized to mark this as, let's say, respiratory system cells, right? And they look different than liver cells, right? Because of the proteins they're out. So we have to constantly be learning these new, they're ours, right? But they're
new proteins each time, a new cell. So so when we start out, we're pretty good at responding to both. Hey, that's self. This has got to be foreign. And then we lose the ability to respond to self. We know what self is now. We don't respond to it. We only respond to something that's different. That's what I'm getting at. Okay? Only respond to something that's different. That way we're not stuck spending too much time trying to identify everything that's going on. Okay. The big deal is when you get sick, that is that primary immune response. Okay? When you are first introduced to that antigen, that's going to be the primary. Okay? So we often say somebody that is allergic to something, they weren't allergic to it before that, right? They suddenly became allergic to it, right? It's not the first time you had it. It's a subsequent exposure to it, okay? So there are people, my father-in-law, he worked in the beehive industry, and at one point he was checking all the beehives in Arkansas. He was one of those guys that had to regulate, had to make sure that you were doing it right and that kind of stuff. So he got stung. Well, he got stung enough to where finally he's allergic to bee stings. And at that point, you lose your job, right? You can't be stung because you're going to die, right? So he no longer did that, okay? There's people that you eat peanut butter. I've eaten peanut butter. I've eaten peanut. Now I can't eat peanut butter. My next door neighbor, bless his heart, he loves plums. He planted a plum tree. He grew this plum tree. The first year, it was not a very big, tall plum tree, but it produced 3 plums. He was so excited. He brought one over to me. He said, he had an allergic reaction to eating that plum. He cannot eat plums anymore, y'all. Right? So what do? I go over and eat his plums all the time right in front of him. Just, oh, this is the best plum. Right? So, the primary immune response says we have to see it before we can have a response to it, right? And in this case, with these allergens, they weren't allergens, they weren't allergens, they weren't allergens. Suddenly they're an allergen, right? Yeah.
Is it possible for an allergen to, I guess, fade away after a while?
Sure, absolutely. And I was one of those. I took allergy shots when I was a kid. And what they're doing is inoculating you with lesser form, small. And when you get a big response, they know, crap, that means your immune system is still fighting that one really, really heavily. Okay? So, we can do some things with that. Okay. But the next time, after you have been sick, been through the primary response, now it's all about the secondary response. You already know how to build this thing. It's already built for you. We just got to go find the right one. Instead of checking everybody to see if we've got it now, we're going to check, oh, there they are. Make a bunch of them, fight the fight. Cuts down on the time completely, to the point where that little bit that gets in your body that normally would be able to crawl into a cell and start making more viral particles and more viral particles and more viral particles, while we're over here trying to fix it, get all the machinery in place, it's going through the infectious cycle. We cut it off almost immediately. Just a few of them get in the body, really before they can start reproducing in the body, we've knocked them out. Okay? So, and the sooner you've had it, the more recent you've had it, is the way I want to say that, the bigger your response is available. Right? Why are we having to get a booster 6 months after you've had your last COVID shot? Because your antibody levels gradually start to wane. They start to go down. And what we want to do is, while COVID is around, we want our system to be way up here. So we reintroduce it. Bam, it comes back up here. Right? So if you've had your COVID shot, if you've got COVID and you're working in a COVID facility,
on the COVID floor, messing with COVID patients, you're cool. Your body is saying, throw them antibodies in all this because it just keeps coming in you, right? So, and you're probably overworked and underpaid and need a vacation now. Okay?
Doc, why would something that hasn't been an allergen suddenly become one?
And my answer is we screwed up. It's just one of those deals. Okay? And it can happen with lots of different things. Certain things are much more susceptible. A lot of times, if we made an antibody that is close enough to something else, we will throw that at it. Okay? So imagine a virus or bacteria that we've made antibodies to, and it matches the allergen close enough where the antibody can bind to the allergen. Then it is bam. Okay? So I think that happens a lot of the time. Or it just could be that everybody hates me, right?
What about when you like had an allergy for something and then you don't?
Same thing. A lot of times it will wear off. That load will go so far down, it no longer becomes an issue. Okay? So, all right. So here's the B lymphocyte. Now notice this one has the D antibodies on the outside of it. So we know it's been built. It's specific to this dude right here, right? Whatever matches this little green Y is what this one is built for. It's one shape, okay? It may not be one thing, but it's one shape. I can say it that way, okay? When we encounter, okay, we check them all, we start checking, and we find this one's the one that works. We're gonna go, okay, bam, you're it. Go through the production of making tons and tons of these antibodies and releasing them. Make that plasma cell, okay? And all of these that came from this one cell, this one just happened to find it. I'm gonna keep some memory cells out there, but all the rest of these that are on this side are all devoted to being plasma cells. I just keep a few of the memory cells back, okay? See that right there? That's a virus. I don't know where that came from. All right, so let's put it together now, because this is what's going to happen in real time. And again, if we were having an essay question back in the day, this would be your essay question. Explain how the B cell and the T cell sides work together in specific immunity. Now there's two slides to this. This is the upper slide, and then there's one underneath it's this lower slide. So we're going to have to flip back and forth a little bit, okay? Up at the top, the little red guys, they're the virus. They're the herpes-gonorrhea virus. That's a bad one, okay? And y'all got it, I didn't, okay? And it infected y'all's cells, okay? So notice that we've got these viral particles in the body. We've got cells making the virus. They're making them, and they're releasing them out into the body, okay? It took over our cell, so our cell became the antigen-presenting cell. It said, they got me, herpes-gonorrhea got me, okay? Exactly. So now, we're gonna go this way, and I'm gonna go out the far, the virus is infected. Number two, the virus and the viral proteins on the infected cells stimulate the macrophage. So let me show you what that looks like. Okay, so all those herpes-gonorrhea viruses that are floating around the body make their way to the club, the lymph node. They come through, bam, you don't belong here, I eat you. Ugh. And then expose it on the outside. Bam, what do I release? Interleukin-1 gets released. That's called the helper T cell. Helper T cell, come check this out. Here's my helper T cell. Let's see what you got, buddy. Show me what you got. Oh, dang, herpes-gonorrhea is in here. Ooh, we've never seen that before. We're going to build cells to get rid of this. Okay? We're going to talk to the B cells and the T cells. So I'm going to talk to the B cells. We're going to say, hey, B cells, y'all come check out. Do we have antibodies for this? No. Hey, T cells, do we have T cells that have the
receptor that can do this. No, we gotta build them. We're gonna get infected. The infection grows and grows in our body, but while the infection is going through this primary infectious stage, we're building the cells to fix it. Once we get the B cell that matches it and can produce the antibodies, we make tons of those B cells through proliferation. And those B cells release as plasma cells, almost screwed up, release the antibodies out into the body humors. They float around and they start knocking out the individual viral particles, right? Okay, they match them, everything's good. That's this side. Here's the B cell, proliferate, keep some for memory, make some plasma cells, okay? Over here on the T cell side, here's the cytotoxic T cell. We get a cytotoxic T cell that has the ability now to identify that antigen being presented to it on other cells. Because that's what it's gonna knock out. It's gonna knock out the cells, it's gonna knock out infected cells like this one, and it's gonna do this, right? And while it's knocking these out, sometimes other viral particles escape that cell as it's being burst or something like that. So we need tons of these antibodies marking these for destruction. Does this kill the virus? No. Does it make it unable to infect other cells? Yes. And do immune system cells know to knock out an antibody attached to an antigen? Yes. So we get it all cleaned up. Okay? So we get it all cleaned up that way.
Can we go to, are you done with this now?
I am.
Can you go to the previous slide, the other slide?
But that one.
Yeah, when you were discussing this, you didn't say anything about interleukin 2. You didn't.
Well, it's right here. It's just,
Yeah, and you didn't factor it in your discussion. You didn't
well, it's,
know.
it is once we identify the cells. We have to come on. I just forgot. Okay. Let me talk to y'all about AIDS. Okay? This is going to be a mini case study for us. Okay? So, HIV hit the United States in 1981, and in 1981, I was in the 10th grade. I was a sophomore. Okay? And what was going on before 1981? Let's say that would be the 1970s and the 60s, and what do we think about the 70s and 60s?
Free love, baby.
Free love. It was free love.
Free love.
So, and that was that in 1980. It is still, and in 81 there was, and all of a sudden HIV hit. No more free love, y'all. I was right there. Right?
You missed your chance.
But you're just finishing your high school.
Well, I just starting high school pretty much, right? So, HIV. HIV is the human, let me say AIDS first. Acquired immunodeficiency syndrome is the disease that HIV can produce, okay? So, the typical infection cycle is we've all got specific cells that are CD4 positive markers, meaning we have the CD4 protein on our cells. Okay? And it's a pretty ubiquitous one in our cells of the immune system, where 60 to 80% of those have the CD4 marker on them. Okay? So, this is the marker that is used by HIV, okay? If we don't have T cells, we can't defend ourselves. So, the HIV virus targets in on the CD4 marker. Just like we said, COVID's gonna target in on respiratory system cells, like hepatitis would target in on liver cells. This one's specific to immune system cells that have the CD4 marker on it, okay? And the way it works is the HIV particle, the virus, has a glycoprotein that's called GP120. And GP120 is like this little egg-shaped and it fits into the CD4 just like that. And that's what allows the virus to dock onto our cell. Okay, it's how it attaches, kind of like the antibody would to an antigen. So, any of those CD4 positives are at risk for HIV with its glycoprotein 120. Okay? After the virus docks, there is an adjacent protein called CCR5. They're kind of tied together. Okay? So we'll do it this way. If, let's say this over here is the CD4, what we're going to bind to, next door is CCR5. Okay? And CCR5 is the hatch that the virus can pop open and crawl into our CD4 positive cells. Okay? So, it's going to be able to move in, and when it moves in, that HIV virus sheds its protein coat. Okay? And inside of this HIV virus, it has RNA. That's really, oh, I shouldn't say it that way. It's RNA, that's its genetic material. Okay? It's got enzymes in there as well. Okay? One of them is called reverse transcriptase. We talked about DNA polymerase making more DNA. If we have one side of the DNA, we can make the other side of the DNA with DNA polymerase. We had RNA polymerase that if we have one side of the DNA, we make the RNA. RNA transcriptase goes backwards. It goes from RNA to DNA. Okay? It goes from RNA to DNA. So, the sneaky little virus has RNA and the reverse transcriptase enzyme to make it produce DNA. If you give us single-stranded DNA, we'll make the copy to the DNA with DNA polymerase, and now we've got double-stranded DNA that is in our nucleus. Okay? Into the nucleus, and that DNA supersedes everything else. And the instructions that are found on that double-stranded DNA that originally came from the virus is how to make more viral particles. Okay? What we're going to do is we're going to do our normal way with DNA. We're going to open it up and we're going to make copy after copy after copy of that RNA. That's the viral RNA. Okay? When we send it out to the ribosome, the instructions for making the protein coat of the virus is found in that RNA. So we're going to do what the cell normally does. If you give it this, it makes this, right? It's like a cookbook. It knows how to follow the ingredients and the directions on the cookbook. So that cell no longer does what it's supposed to be doing. It is just a virus-making machine now. Okay?
For HIV.
Yes, for that particular virus. And I've told my classes this for a while now. Y'all understand how a computer virus works, right? Where you open up an attachment, it infiltrates your system. It then takes over your computer to then make more copies of the virus and distribute it out to other computers, right? Okay? That's perfect when you think about it. That name computer virus comes from this system though. This is what I learned. I learned it this way, and then when computer viruses came online, it's like, oh, that's awesome how this works. I mean, it's doing the same thing. So, once we build enough of the virus, kick them out of the cell. Okay, here's what it looks like. Here's an HIV virus. Okay, here's the protein coat that goes around it. Here's GP120, that glycoprotein that we'll end up building for it. Inside of it, you can see the genetic material, some enzymes and stuff like that. This little red one here is CD4. Pink one beside it is CCR5. Okay, so up here, notice that we've got the virus attaching to the CD4 marker with its GP120 glycoprotein. Okay, once it attaches, now CCR5 is the way in. Pop the hatch on CCR5 and crawl in. When it crawls in, it's going to release okay, shed its protein coat, jump in, notice that we've got the viral RNA, that's in green, and we've got the enzyme reverse transcriptase in blue. What the green makes is the blue, DNA now. From RNA, we're gonna go to DNA, okay? So every time it sees a U on the RNA, it's gonna put an A, right? So, we end up with the DNA strand. We can make a copy of that using DNA polymerase, okay? So we make a perfect copy, a complement to it. Move that into the DNA of our host cell, incorporate it in, and now this blue part supersedes everything else. We're gonna use our RNA polymerase to make RNA, send it out to the ribosome's gonna read it, make the proteins that we want it to make, use this green as new viral RNA, put it together, ship it out of the cell. Sneaky, sneaky, right?
Wait. Yeah, when you have the CD4 cell down there, and the viral exit by cells, lysis, and T cells, you didn't experiments like this.
But it can leave this, it can leave the cell by either exploding the cell or through exocytosis. There's two ways that the virus leaves the cell. It
So
can
it
either
means
leave,
it's no
it can
longer-
either leave as exocytosis, where we just release it from the cell, or we build enough of the viral particles inside of it, it bursts
So
and
does it-
kills the cell.
Does it mean at the point the viral is exiting the cell, does it mean it's no longer replicating? Is that what, does it mean this succeed in replicating?
That means it's just, it's a new virus that's out there in the body.
What I'm saying is this, at the point it's exiting, does it mean it's no longer replicating? Because it means, because from this diagram, from,
Once it exits here, now it's able to go out and infect another cell and reproduce again.
in the same human being. Oh, wow.
Or I can give that HIV can be given to somebody else, and in that person, the virus can then start the infectious cycle.
Okay, I thought when we say exit, I thought it means it will not stop. That means the person will be free from the effects of the
No,
viral infection.
that's how the viral load continues to go up in the person. Okay,
Doc, is that how all viruses proliferate?
pretty much, pretty much.
Can you go back to that picture and then go over what CD4 and CCR5 are?
Yeah. CD4 is the little red protein receptor that we have on it.
Just on one of our cells?
Yes. And 60 to 80% of our T cells are CD4 positive, meaning they've got that protein. And then the CCR5 is just another protein right beside it. And that's the hatch to get in.
So it has to ride on CCR5 to get into our system.
It has to get in, it has to attach to CD4, and then it has to pop open CCR5 to get in.
And then the
So is it
GP,
the 64? Sorry.
the GP120 is just a glycoprotein for HIV?
Yes, it's what matches to the CD4,
Okay.
GP120.
So it's the CD4 that pops up the CCR5?
No. CD4 is the marker that it attaches to, so that gets it there, and then CCR5 allows the virus to get in, because it has a key to get through that protein.
If GP120, the glycoprotein, attaches to the CD4, it binds together, right? So when it binds together, for it to get into, for it to be able to replicate, it has to attach to CCR5, or is it the opening, the entrance, or is it the cell that acts as an entrance
It's the entrance.
to get into our system?
Okay, so when this happened, one of the first drugs that we tried on it was AZT. And if you watch Dallas Buyers Club, this is the one that Matthew McConaughey, all right, was working to get. They were trying to get the drug companies to go ahead and test on humans because they were dying so rapidly. They're like, at least give us a shot. But they
Food and Drug Administration was dragging their feet, trying to do all these tests and things like that. And the community that was being, that was infected, went out on their own a little bit, okay? And actually got some good stuff done. Then we started using protease inhibitors, okay? So what AZT did is it shut down the effects of reverse transcriptase, okay? Protease inhibitors inhibit the making of the proteins, okay? So, there was an issue though. HIV also does viral shifting, antigen shifting on it, like the flu does. That's why we have to have flu shots every year, right? Because the antigens shift on the surface of proteins, like what's going on with COVID, but not as great as it does in the influenza virus, okay? COVID's been shifting. We've gotten, what, 5 strains now that they're working with? So, and so far, our vaccinations have matched, okay? All right, so we use combination drug therapy. I was teaching a class in nutrition and talked about AIDS. It showed 2 men that were HIV positive, and the drugs that they took back in the day when all this was coming out cost them, I think it was $65,000 a year, and it was over 100 pills a day that they were taking, okay? And now we've gotten a lot better, and the drug prices have come way down and that kind of stuff, and the goal is we can't cure this, but we can manage it, right? So, try to keep the disease in check. We've been able to reduce the death rate this way by 75%, okay? When it first came out, you didn't know if you could hold hands with somebody, right? If you could kiss them, if you sat on a pot behind somebody, I mean, there was no idea. So, there have been people that have gotten a weaker form of HIV, okay, so they never develop AIDS, but they are HIV positive. So this was something, could this be a treatment plan? Now that there are antibodies to this, would it make them immune to getting a more virulent form of HIV? That's been kind of negative because you'd have to tell somebody, well, I'm going to give you a lesser form of HIV. Hopefully it'll protect you from HIV. So that didn't catch on very well. We've got drugs that will block the actual binding sites, okay? So that the HIV can't get in. And,
Excuse me, sir.
Mhm.
At this point, the one the blocking, the one that can block the binding site, is it preventing the GP 120 to bind to the CD4?
Yes.
Or
Yes.
okay.
okay, and then we did find that there are people that are completely immune to HIV.
Really?
Okay, there's 10 to 11%, okay, of Caucasians, less than that, about 2% in African Americans that possess a mutation in their CCR5 protein. Okay, so imagine that there's 1,000 bases to make the CCR5 protein. Okay. And these individuals that had the mutation, they lost 32 of the 1,000 bases. So that means that they don't have several amino acids are missing. That means HIV doesn't have the key to get in, okay? It can bind to CD4 just fine. It can't get in through CCR5 because it's a different shape.
Yeah, on CCR5.
The people that have the mutation have a mutated CCR5 and the HIV virus can't get in. Different lock, okay? It's got the key to the regular lock. This is a new lock, okay? So the question always is, well, can these people give somebody else the HIV? And more than likely, since this is a virus and it can't proliferate in the body, there shouldn't be no infectious cycle here, right, where it actually ramps up in the body, okay? But eventually, it's foreign. The body should not get out, okay? So it's just kind of cool. And this I always think about I Am Legend then, you know, where there's somebody that makes it, right? And that's really the way most immunologists believe. There's not going to be this plague that wipes out mankind completely. There's always going to be some people genetically okay? Or maybe they made an antibody differently that they're gonna be able to survive. It's kind of- All right,